Palestina Guevara-Fiore, and they are going to talk to us about noise pollution affects sexual behavior but not sperm quality in guppies. So welcome, Palestina. And
Hello, can you hear me?
yes, we can hear you.
Great. Let me share my slides.
Yes.
Perfect. Can you tell me if you can see them?
Um, we are waiting for. Okay, yeah, we can see the slides.
Perfect. Thank you very much
Yep.
to you and to the other organizers for this conference. It's great to see all the talks and it's a great effort. Thank you. So today I will talk about noise pollution in a model that you probably know, the guppy. And this is, of course, not my work alone. I work with, my slides are not changing. Give me 1 second. Don't know why.
Yeah, sure. Take your time.
Cheers. Uh-oh. Okay, here we go. Okay, so this is our team. The lab work was done by Jimena Zavala and Tania Lopez, where they did their honors thesis in my lab. This is Alejandro Rios, who is an expert in sound. He usually works with birds. This is his first aquatic research. And this is Rosalina Reyes. She's an expert in sperm quality. And Felipe Pacheco, who is a physicist, and he helped us to measure the sound in water, which was a challenge. So we live in 2024. Everyone is familiar with pollution. And recently, we have realized that noise is a pollution that affects our own health, but also affects other living organisms. Sound is a propagation of waves through an elastic medium such as air, water, or solid materials caused by the vibration of an object, which then transmits energy through the medium. So, and in biology, noise is the unwanted or disrupted sound that interferes with normal biological functions. For example, it can affect communication, behavior, or physiological processes. And the source of noise can be natural, so imagine the wind or the water moving in a river, or it can have anthropogenic origins. And we know it has effects on animals. The research of noise pollution has mainly been conducted in the air, in terrestrial animals, because in water it's more difficult to measure. I think that's the reason. In water, the sound travels faster and longer distances, and it has better propagation. And aquatic animals like fish can detect sound because they can detect pressure sound and particle motions. They have an inner ear and a swim bladder and Weberian apparatus. So they can detect sound. The auditory ranges in fish overlap with anthropogenic noise, and that's why it's important to study this because it does interfere. Indeed, noise has effect in the physiology and in the behavior of many fishes. It can change the stress levels in some fish, the development and their growth, and the auditory threshold. And in terms of behavior, it can change their social foraging, anti-predation, and social distribution, and also sexual behavior. And this has mainly been studied in fish that use acoustic signals to communicate, but we don't know much about fish that don't use acoustic signals like guppies. So we decided to ask how does noise pollution affect sexual behavior and sperm quality in Poecilia reticulata, which is a model system in evolutionary biology and behavioral ecology. Now, these fish don't use sound like that, or not. They don't use it very much, but they do get noise from the environment. In Trinidad, you are very familiar maybe with the waterfalls, and that's a sound source. And also in the rainy season in Trinidad, the rivers get bigger and they are very, very noisy. Anyone who has had an aquarium at home knows that these air plumps are really noisy, and this can also have had implications for the fishing in aquaculture. So guppies are live bearers. They have strong sexual dimorphism. They use this little anal fin called gonopodium to inseminate females. And they have been studied a lot, particularly related to their sexual selection. So what did we do here? We conducted 2 independent experiments, one to test the prolonged effects of noise. We tested noise, we gave noise to fish, white noise, for 4 days, and we recorded behavioral trials, and we registered the sperm quality of fish before and after 2 treatments, noise or the control without noise. And we conducted another experiment to test the immediate effects of noise. We gave a single exposure to noise to males, and we tested their sexual behavior. So for 4 days, we tested behavior and sperm quality, and we tested the behavior of males when they were listening to noise. So in terms of long-term effects, we gave exposure to treatment or without white noise for 12 hours over 4 consecutive days. It looked like this. We set the fish in their experimental or the treatment tank, sorry. We let them acclimate, and then we collected sperm samples, and we measured their behavior before and then after 4 days of treatment. The treatment could be noise or no noise. This is our experimental setup. In these individual aquariums, we place each male, and we measure the noise using an hydrophone and also an accelerometer. And each fish, the males couldn't see each other, but around these little tanks, we had females, so they could see females. So they could see females swimming around them. In terms of the immediate effects, this is real-time exposure to noise, and the control treatment will be without noise. And what we did is we had 1 male with 2 females, 1 big and 1 small, and we recorded their behavior for 30 minutes. What did we record? For those who are not familiar with guppies have 2 ways of inseminating females. They can court. Here in black is the male and in gray is the female. They can perform a sigmoid display in front of the female, and that's what we call courtship. Or they can have a sneaky behavior, which is they thrust their gonopodium into the gonopore area of the female, and this is forced copulation attempt. And we measured the latency, the frequency of gonopodial swings, frequency of gonopodial thrust or forced copulations, frequency of sigmoid displays or courtship, and the duration of the sigmoid displays. And with those data, we could measure 3 different things: the reproductive effort, which is the behavior that they do to both females, or mate choice, which is the behavior that they directed to one female in particular. And in poecilid fish, males prefer larger females. So that was our prediction, that they will go for the larger female. And then their mating strategy prefer courtship or forced copulation attempts. In terms of the sperm quality, we measure number of sperm bundles, motility, viability, sperm concentration, and DNA damage. And guppies are widely used to test the sperm quality. This is a picture of the sperm bundles. Sperms are grouped in these little bags called sperm bundles, and you can measure all these things, which we did here. So in terms of long-term effects, after being in 4 days hearing noise, we had no effect on their behavior in terms of latency or the difference between frequency and duration of courtship. But we found that the reproductive effort increased after noise treatment. And we had no effects on sperm quality. So in these graphs, we have in the x-axis control, the treatments control on the left in yellow, and noise on the right in orange. And in the y-axis, we have the difference between after and before. Remember, in the prolonged experiment, we gave 4 days of noise, and we measured before and after. So what we found is there was no difference in the frequency of courtship, duration of courtship, but we found that the reproductive effort increased in the males that perceived the noise for 4 days. And I repeat, we had no effect on sperm quality. I'm not going to present the graphs, but we couldn't find any difference between the samples before and after the noise. So the effects on sexual behaviors, the frequency and duration of courtship remain constant. So what could have happened? Maybe they have insufficient visual and olfactory stimulation during the treatment, or maybe they had unnecessary stress due to isolation. But what we found was an increase in reproductive effort, and what's increasing here is the frequency of forced copulation attempts, the sneaky behavior. So this is not significant, but when we measure reproductive effort overall, which is the number of courtships and the number of forced copulation attempts that the males are performing, we see a difference. So it seems that males are doing more forced copulation attempts. So it's likely that this tactic increase under acoustic distress, which is in accordance with the literature that says that forced copulation attempts perform in males that are stressed, and also that courtship involves more energetic costs, and then the males prefer doing forced copulation attempts when they are under stress or energetically deprived. So the effects on mate choice and mating strategy, we know that results show that noise does not act as a stimulus capable of altering generalized preference, because we found that there was a preference for big female. So as I told you before, in this family of fish, males prefer the bigger one, which it seems to be related to fecundity and therefore fitness in males. In terms of the sperm quality, there was no significant effects of noise on sperm quality parameters. It's possible that they had an habituation to stress, that this could have occurred. For example, in our aquaculture facilities, the fish had a filter and an air pump. Of course, we measure the noise between the treatments, and there was a significant difference of noise. but still, these fish were in the lab a few months before the experiment. They were wild guppies from our city, Puebla, but they maybe got habituated to the noise we have in the lab. It's also possible that noise can be a stressor that doesn't leave a physiological trace, and this opens a window for future research. Now, it's also possible that we didn't give enough time of noise, but we follow also the literature and also the sperm production. If in guppies 4 days is enough to get changes in sperm production, so we don't know what could have happened, or maybe guppies are just the noise is just not affecting them, which is also possible. As I said before, in their environment, they are used to strong noise from the river, even though they don't live here. They live at the edges. They have exposure to noise. So in terms of immediate effects, which we only measure in behavioral trials, there was no effect on latency, sexual behaviors, reproductive effort, mate choice, or anything else. So if you play noise to a male that is courting females, it won't affect it. So in conclusion, the results show that noise exposure does not have an immediate effect on behavior. It causes a significant increase in reproductive effort, which is primarily due to an increase in number of forced copulations. It does not significantly impact the measured parameters of sperm quality. So we can conclude that noise-induced stress is moderate in this species, primarily affecting male sexual behavior, but the long-term consequences of this change are unclear to us. So I would like to thank my lab members, and I'm very happy to answer any question you may have. Thank you.
Well, that was a really great talk and also some clear results. So you already have a bunch of questions. So first question is from Alexis, and he's asking when the noise was displayed during the day.
Yes, the noise was displayed during the day. And what we did is we had some gaps of noise. So because the noise could damage the ear of fish. So it has been shown in the literature. So what we did is we gave some minutes of noise, and then they rested 1 minute, and then again other 5 minutes. This was done randomly, so it was not constant. And then the fish were resting in the night. Yeah.
So next question is from Law, and they're asking, nice talk. I might have missed this information, but what kind of noise did you use? And do you think you could have a different behavioral reaction to a new noise, something they have never heard, versus a normal pollution noise?
Yeah, that's because of time I didn't mention this, but we use white noise, and this was artificially generated with Audacity, and what we use in the literature, white noise is used usually because it covers a range of frequencies that cover all the fish, all the fish auditory input. So this white noise oscillates between 20 and 20,000 hertz, which is the audible sound spectrums of humans. And these also include similar fish to guppies. So we know most certainly they can hear it. And we chose white noise because it's usually related to anthropogenic noise.
And since we are running out of time, one final question by David. Is it likely that the effect on guppies will be representative of many other fish species? Do hearing ranges vary?
Sorry, the last part through what?
Do the hearing ranges do they vary?
Oh, yeah. Okay, that's a good question. Of course, when we what we wanted to do is use guppies because no other fish uh, that doesn't use sound to communicate has been used before. But also because guppies are similar to many other small fishes around. So I believe that this could be extrapolated at least to porcelain due to their phylogenetic relatedness. But yeah, I don't want to say it's completely general to other fish. But I don't know. I would say yes, you can extrapolate, but I don't know what we have seen in the literature because this is really new. What we have seen in the literature is fish that use sound get affected by noise, just like birds that they communicate by singing, and then anthropogenic noise disrupt those signals. So in that sense, fish that communicate with sound they get disrupted too. In our case, we didn't see that. We didn't find that this was a strong stressor. But we don't want to say noise pollution is not important. I think uh, we set the first stage to test it in a wider range of fish. Yeah.
All right, thank you again for your great presentation and best of luck with your future research.
Cheers.